So this is the long title I have chosen and scared Monica because she thought I was not going to talk about what she wanted me to talk. So I would like today to go through the reproductive cycle of grapevine. So we start very early. We're going to start from the differentiation of the inflorescences, and we'll end up with the ripening process. And at the end of my talk, I have just slides for a 5-minute discussion on what we think are the processes during development and ripening that red blotch interferes with, resulting in the changes in berry composition at harvest due to the virus. So to start from the very beginning, for the basics of the topic, when we talk about berry, we refer to a very specific botanical term. Berries are fruit with a typical fleshy pulp called mesocarp, fleshy pericarp called mesocarp, that derives from the development of the ovary, the female part, one of the female parts of the flower. So we cannot talk about fruit without talking about flowers and their development. This is, you know very well, this is a hermaphroditic flower of a grapevine, where you can see in this particular slide I focus on the female part. We call the female part the pistil, that comprises a stigma, a style, and an ovary. We're going to see later that the stigma is very important because it's the part of the pistil that receives the pollen from the anthers. The style is that structure that guides the pollen tube toward the ovary, and the ovary obviously is where fertilization happens and ripening, development and ripening starts. Male parts, also important, because without fertilization you don't get fruit. You have the anthers, the distal part of the stamens, where you mature the pollen grains. Pollen grains will be released at anthesis, at cap loss. Cap loss will be released, end up on the stigma, and everything starts from there. Okay, so these are the flowers, just a refresh of their anatomy. And this is something particular in plant biology. For grapevines to make flowers, it takes 2 years. So we should not forget this. We start from those compound buds. We're going to talk in a little bit. In those compound buds in year 1, we differentiate the inflorescences, these microscopic primordia, early organs that don't look like inflorescences, but they will become inflorescences. We finish year 1 with dormancy. So those buds where the inflorescences have differentiated, we enter dormancy. We start year 2 by bud break, and during year 2 only we're going to have the development of the flowers, and finally fertilization and fruit set. Okay, so it's a 2-year process. So when we think about yield in grapevines, we need to think about all the processes that occur in the 2 years, starting from the inflorescence differentiation in the compound buds. So when you think about the yield per vine, our final yield, we need to think about the number of clusters we differentiate in each bud, how many buds we keep on the vines at pruning. So we're going to talk a little bit about how inflorescences differentiate and the number of inflorescences that differentiate in those buds, how this is determined. Buds per vine determined by pruning. Obviously, we keep a certain number of buds with inside them a certain number of inflorescences. And then we go in year 2. In year 2, we're going to have the number of berries that actually set on those clusters. And then we're going to have their development and ripening, influencing obviously yield and composition. So if we look at this is an equation that spans the 2 years of the reproductive cycle of a grapevine. And we're going to see that the yield potential is defined in year 1. So the number of inflorescences you differentiate define how much maximum we can achieve in year 2. So there are a series of processes very important that happens in year 1 which will influence yield in year 2. And we're going to see later on that actually what happens in year 1 is the major cause of yield variability across the years in a particular site. We're going to see that this yield potential accounts for about 70% of the variability in yield, whereas berry cluster and berry weight will account for 10 to 20% of yield variability. So that's why I wanted to go through the reproductive cycle and not only focus on what happens in development and ripening, which is typically what we study in our labs, but is also the result, is very, very tightly linked to what happens in year one. And also is a good reminder for viticulture is that whatever we do to the vines in year one will affect year two at the final yield. So let's start from here. From after bud break, you see your primary shoot developing from your compound bud. And here you already have the complexity of the overlapping 2 years of reproduction. So if you look at this shoot, you're still seeing those clusters. Those clusters are typically in year 2 phase of the reproductive cycle. They will differentiate flowers and berries at one point. But at the axil of these leaves, you start differentiating the inflorescences, which will develop in year 2. So if you look at this, we have clusters representative of year 2 of the reproductive cycle, buds representative of year 1 of the reproductive cycle. And this is something always to keep in mind, that you have overlapping things happening at the same time in the vineyard. So as you are differentiating those flowers, you are also differentiating the inflorescences for the following year. So for example, if you're now trying to push a fruit set, you are a table grape grower, you're trying to push fruit set by adding gibberellic acid, for example, you will spray those clusters a little bit later than here, but you will spray those clusters, and you may actually not only affect the fruit set, but also affect what happens in the buds, with consequences on the yield in the following year. And we're going to see how gibberellic acid may actually be a negative regulator of what happens in those compound buds. Okay, so can you hear me? Follow me? Good, good. Okay, so let's go now a little bit deeper in what happens in those buds. So if you look at every leaf on a grapevine, you're going to identify 2 buds, a lateral bud and a compound bud. Lateral buds produce those lateral shoots that develop the same year the buds are made. So the picture was taken too late. A lateral bud already pushed a lateral shoot. And here at this axil, you will find a second bud called compound bud. Okay? So compound buds, and now here I wrote down some of the main features. So lateral buds will push a shoot the same season they are produced. Compound buds have a particular feature, which is they become dormant at the end of the season. And the compound buds are typically the fertile buds that next year will push shoots carrying clusters. So we're now focusing only on compound buds. If we look now with a microscope in those compound buds, we'll identify a particular structure called the shoot apical meristem. This is key in every plant we know of for their growth. This is a meristematic tissue or a stem cell type of tissue where you have a central zone of cells that don't do much besides keep dividing. They are undifferentiated, so they all look alike. They keep dividing, and as they divide, and the cells that are generated by the cell division will move along the apex, they will start producing organs. Okay, so this keeps dividing and growing, and now you have this LP stands for leaf primordia, the formation of leaves on that particular shoot apical meristem. Okay, and I think a video is always better than words. This is a model, a computer model based on mathematical modeling of the growth of a shoot apical meristem. So here I have the apical meristem dividing, and these are the organs that form as the shoot apical meristem divides. Okay, so this is not grapevine, this is Arabidopsis, so the positioning of the leaves is different, but just to give you an idea how this process proceeds. So your shoot apical meristem there, leaf primordium, etc. Okay, so this is what is happening in those tiny compound buds. You have the shoot apical meristem making new leaves. And this is the typical what happens in grapevines. You have leaves at opposite sides. Here you're going to have your apex dividing and leaves being produced. But you also know that we don't only make leaves on grapevines. We also make clusters and tendrils. So a particular shoot apical meristem in those compound buds will typically divide, produce leaves, and at one point produce those uncommitted primordia, we call them, or anlage, which are lateral meristems, so a lateral structure that can differentiate into either tendrils or inflorescences. So here is a turn point. The plant is given the possibility to enter reproductive growth, but is not committed yet. And that's, with my students I always talk about the decision of making kids. This is something that you should think about, and And as a vine, or as a person, you may think about your bank account, how much you have in the bank, and a vine may think about how many resources has stored in the vine. Is it time or not to enter reproductive growth? Am I stressed? And for a vine will be water and pathogen stress, for example, at that point. Maybe it's not a good time to enter reproductive growth. Let's keep growing vegetatively, make tendrils and keep growing, and maybe next year will be a better year to have kids. So that's your uncommitted primordia. And we're going to see that these uncommitted primordia can turn into clusters or tendrils. Tendrils are particular structure that you know will help the vine to grow on another structure, and we call them young clusters because we can actually convert this tendril into a cluster by applying specific hormones. So they are developmentally related. And this is what I'm talking about. So this is actually a close image with an electron microscope of an actual shoot apical meristem inside a compound bud. So here you have your shoot apical meristem dividing, a leaf primordium being developed, and then a tiny uncommitted primordium. We give the chance to go or not to reproductive growth. Here a little bit later, you're going to still see your shoot apical meristem growing, a leaf primordium, and the uncommitted primordium now is expanding. Now it can go both ways. Depending on how many times it branches, it divides, it can become a tendril or an inflorescence. So this is a turn point here. Either you become a tendril or become an inflorescence. So this structure here, again, is a primordium, is a microscopic structure that carries all these little tiny parts which will become your flowers the following year. Okay? So the number of inflorescences that you will generate in a single compound bud depends on the decision that the shoot apical meristem makes to become either an inflorescence or a tendril. And this is, we know something about it, and we know some hormones are involved in this. Hormones are those molecules that have very low concentration inside the plant that are produced by plant cell determines developmental events, in this case inflorescence differentiation. So typically you have your shoot apical meristem. Gibberellis actually promote the formation of those uncommitted primordia, but then they become a negative regulator of inflorescences. So remember before I said you spray with GA a young cluster in order to improve fruit set and early berry development. But at the same time, the gibberellic acid can have an opposite effect in reproductive growth inside the buds. So you may promote year 2 yield, but you may actually inhibit year 1 success. Okay? And we know something about the positive regulator, which are cytokinins. Actually, we can treat a tendril with cytokinin, and we can make a tendril become an inflorescence. So this is something about the regulation, and this goes beyond obviously the endogenous regulators. We not only have hormones that regulate this, but also environmental factors. Again, let's go back to the analogy with having kids. The vine need to have not to be stressed. So water and nutrients are a clear message to the plant if it is time or not to make kids, to make inflorescences. Water and nutrients, temperature and light. Low temperature and low light exposure of the shoots that carry those buds typically reduces the conversion of those uncommitted primordia into inflorescences. It pushes more the formation of tendrils. And all this is interpreted by the plant, the internal hormones, the nutrient status, water status, light, temperature, in determining whether it will go through the formation of clusters of tendrils. So remember before I mentioned that the yield potential, depending on the number of clusters we make in year 1, account for 70% of yield variability. This is the reason. We have so many factors that influence whether or not we make inflorescences and the number of inflorescences we make in a bud, that imagine you are now in a cold climate. It's not the case of Napa, but you're in a cold climate viticulture area. Now light and temperature may become limiting factor one year and not the following year. So one year you have low light and low temperature at the phase of production of the inflorescence primordia, and you will get 1 or 0 inflorescences per compound bud, and will have affected by 50 or 70% the overall yield at the end. Okay? So this is just to show the complexity of the process. We know a lot about it, and it can be very important in determining your yield, and that's what I'm talking about. cluster per bud, buds per vine. This is you deciding how much you prune. This is the vine deciding how many kids he wants to have the following year. 6 to 70% yield variation. This is a study where they looked at yield variation across multiple sites with multiple varieties, and 60% of the variability is accounted by the number of bunches you produce on each vine. And when they look at the other factors, berries per cluster, berry weight, 30 and 10% of the variation. Okay? So what happens in year 1 has a real effect, impact in what is your yield in year 2. Okay? Okay, so now we have made those inflorescence primordia, or not, we've seen that may not happen, and the buds enter dormancy. So this is the way the vine can survive winter cold, by forming this very specialized structure that are protected from dehydration, they're protected from cold, they're protected from pests and diseases. And if you cut the bud, you will see something that looks very much like the shoot apical meristem I showed you before, with your leaf primordia. Now they look like more like leaves. And then if you look at the axil of some leaves, you will find those inflorescences primordia. So a way to actually determine how many clusters per vine you will have the following year is by dissecting buds, by looking how many inflorescences you have differentiated in that particular year, and whether or not they're viable at this stage of dormancy. Okay, so now we start year 2. We're done with year 1, we start year 2, we have to exit dormancy. And you know that vines, the first thing that tells you that spring is coming and growth is coming is bleeding. Okay, vines they don't lose blood, obviously, they lose xylem sap. Okay, so this is a liquid made mostly of water, where there are proteins, there are sugars, and there are hormones inside. There is cytokinin, for example, in the xylem sap. And this typically is a way of the root system that is perceiving the warmer temperature in the soil to tell the buds up there to push new shoots. So the water is pushed throughout the vine, these establish the xylem conductivity and send messages, cytokinin to the buds. Now the buds, thanks to the roots, understand that it's time to grow, and they start growing. And when they start growing, now those differentiated inflorescences will start developing the flowers. So only at this point we start seeing flowers inside those compound buds. And this is not a grapevine flower, but just to remind you of the different structure you normally see on a flower. So on a flower you see the first order is the calyx here, made of sepals, typically green, not colorful like the petals. And then you have the reproductive male and female parts. In grapevines we don't see those structure. We know that the petals are lost at anthesis, at bloom, at capfall. The sepals, we don't really see them unless we take a microscope or we take a hand lens. So, but if we look at the very beginning of flower differentiation, sepals are there. So this is your apical meristem of the flower, and you start forming all these structures you see typically in flowers. These guys here are sepals, and these are the beginning of your petals. The sepals grow so fast at the very beginning that engulf completely the flower. So you actually don't see the petals, don't see the stamens, and don't see anything else than just the sepals. At one point, obviously, they will stop growing, and the other parts of the flower will become predominant, so that at the end we don't see actually any of this structure here. So here are the other cool micrographs showing again the growth of the flower. Your sepals growing, and these scientists actually broke the sepals to see what is inside, and you can find the petals, the corolla here, and inside the stamens. Now the internal parts are pushing through the corolla, and at one point you will start seeing the petals coming out, and the corolla, the calyx, your sepals will just disappear. At the end of the day you will have a structure which is very much the flower that you know well of. Your petals fused at the tip, your stamens inside, and the female part. This is a very nice image of the internal parts of the female part. We have again your stigma, style, and the ovules. Here is where the seeds will form after fertilization. And just to remind you, cap fall refers to the loss of the petals at bloom. So the petals are very particular. Normally petals are attached to the base of the flower. So when the flower opens, the petals stick to the flower. In grapevines, the petals are fused at the tip. and not very much at the bottom. So when the anthers now are growing and pushing out, they will remove the cap, and that's why we have cap fall at bloom. And that cap fall process indicates that the stamens are now mature, they have produced viable pollen, and they are releasing the pollen on the stigma and initiate the process of fruit set. Okay? So, and this is what I'm talking about. The calyptra made of petals is now being pushed away by the growing stamens. And as they push, they release the pollen, and pollen lands on the stigma. And this is where everything starts in terms of fruit development. Here you can understand this is a very efficient process. You're now losing the cap, your stamen are very close to the stigma, and the pollen does not have to go very far to find the stigma. And that's why hermaphroditic flowers were selected when we selected vinifera variety during domestication thousands of years ago. So as these grains, pollen grains, land on the stigma, we say they germinate, which is not actually germination of a seed, but is a production of a structure called pollen tube. This is a very fascinating process where the stigma, so the female part, is giving water and nutrients to the grains, to the pollen grains. The pollen grains perceive the stigma, perceive the female part, and start growing. And they grow very smartly toward the ovule. Not because the pollen is smart, but because the female is telling where to grow. And we're going to see, I have a video about that. So, but this is what happens. So you have this sticky, wet stigma that feeds with water and nutrient and signals those pollen grains. The pollen grain will develop into a pollen grain tube and look for the ovules where it will initiate fertilization. And this is the process that without which we don't have seeds, and is the process that activates a downstream series of events that will result in the early development of the berry. So if you are a table grape grower and you don't make viable embryos, because you're growing seedless varieties, you will need to compensate for the lack of these events, which typically happen in seeded wine grape varieties. This is just a cool video. It's not from my lab, but it's just to show how dumb is the pollen grain tube, but how smart is the female. So this is you are in a petri dish here. So this is a growing pollen tube, and you're going to see, I'm going to show you, this is a pipette tip. Depositing one of the molecules was identified as a signal that females send to the male to direct the pollen grain, the pollen tube toward the egg for fertilization. So you can see we can now confuse the pollen tube. So we put it there and it turns. So that's how the female, by releasing these lure peptides, tells the pollen grain where to go. So when you think about this process, so the female wants success in fertilization, obviously. But it creates the condition so that only the smartest will get to fertilize the ovule, the egg. So it needs competition. So the more pollen goes on that stigma, the better the berry will develop at one point. The better signals will result in the fruit set and berry development. So that's what happens. So now we have fertilized the egg with the pollen tube, and the ovary, remember the base of the female part now starts growing right away after fertilization happens. And this is a very early fruit set, so the stamens are still there, but they will dry out and fall off. The male has done his job and has to go. The stigma now is not that wet nutrient structure anymore, it's drying out as well, it's necrotizing. So at the end of the day, you will just find the ovary. The ovary is now developing into a berry. Okay? So, and we know a little bit about the signals that trigger these early events of ripening. So now we have of berry development. The ovule, the unfertilized ovule, is not doing much in terms of convincing the pericarp to grow. So this is in no growth. Before fertilization, the ovary wall stays like that because there's proteins that inhibit that growth. Fertilize, the pollen grain goes in, fertilize the egg cell, and the embryo, the developing embryo, start producing a particular hormone called auxin. This auxin will diffuse by transport into the pericarp, the wall of the ovary, and induce gibberellic acid biosynthesis, which will trigger the development of the berry, the cell division that is associated with berry development early on. Okay, so here we start with the topic of today, which is berry development and growth. But So you know, you heard about this, probably you saw this slide thousands of times, where we show the particular pattern of growth of grape berries that follows what we call a double sigmoid curve, where you have the initial fast growth, an intermediate stage we call stage 2 of stationary growth, which nothing really happened in terms of growth, and then a stage 3 where we again grow exponentially in size. So that we like to divide berry development and ripening in these 3 stages. And when I talk about berry development, I talk about stage 1 and stage 2. And when I talk about berry ripening, I talk about stage 3. Okay? And specific metabolic and developmental events happen during the progression of development and ripening. So typically stage 1, the berry is still attached to the rest of the vine by xylematic conductivity. Berries are green. They are not yet accumulating anthocyanin, which will change the color of red berries. And they are accumulating, depending on the variety, specific other metabolites, including organic acid. Tartaric acid and malic acid typically accumulate early on, whereas sugars, color, softening happens during berry ripening. And this is, from an anatomical point of view, the early growth, stage 1 growth, is due to cell division. So the number you have in the ovary, you have a number of cells. These cells, as fruit set starts, start dividing and expanding a little bit. So if we now plot the number of cells in the pericarp, so in the ovary wall and in the becoming pericarp, we reach the maximum number of cells in stage 2, which means that 100% of the cells are produced in stage 1 and 2. Which, if you think about what you're doing to those berries before ripening starts, stresses, for example, water stress or nutrient stress, may actually decrease the number of cells in the berry. Okay? Whereas when you stress the berries after veraison, you affect all those processes that involve cell expansion. So you have fixed number of cells determining stage 1 and stage 2, that now start growing, getting bigger and bigger and bigger during the stage of ripening. Here just pictures, because I like pictures, and I think they're clearer than my explanations. So here you have it's still a flower, so this is still the ovary wall. You can see that actually the epidermis and the mesocarp, they look alike. We don't have much difference in size. Difference in size is going to be very remarkable at the end of the process. So at this point, you start dividing. So if you think about this, the berry as a balloon, in order to become bigger, what you need to increase the surface of the balloon and fill this balloon with liquid and cells eventually. So what you do, you typically divide the epidermis, so to increase the surface of the berry. You divide the hypodermis, the cells underneath the epidermis will divide, and that's your skin thickness. The more the cell division you happen underneath the skin, the epidermis, will determine the thickness of the skin. And you know that thickness of the skin determines how many other good things you have in those berries, including anthocyanins. So the skin is expanding, the hypodermis is thickening, and at the end of the day you're going to have something that looks, let's go here, that looks more like this, where you have here you have already the accumulation of color in the skin. Polyphenols have been accumulated in the hypodermis. And now what happens in the pulp is that cells actually divide in every direction and at one point start getting bigger and bigger and bigger. And to get bigger, you need to get water into those cells. And this is what's happening. So the epidermis now is actually the epidermal layer, the first layer of cells remain small. We have divided and formed the skin, and the cells underneath the skin are expanding. So this, if you look at the initial and final size of those cells, these guys will not have expanded very much. The internal cells of the pulp, from the ovary to ripening, will have expanded more than 10 times. Okay? And this is actually in the same scale. So these are the internal cells of the pulp. And very different things happen in the epidermis and happen in the pulp. And very different things happen during the process of development and ripening. Here is just a summary slide. which I think is very representative at least of the primary metabolism of the berry. Here we are in stage 1 and stage 2. Stage 1 and stage 2 we increase acidity, we accumulate malic acid, we accumulate tartaric acid. Then something happens in those berries. At stage 2 we stop making tartaric acid, we start using malic acid. So the result is that now acidity declines. At the same time we stop making acids, we start importing sugars from the rest of the vine. Okay, so this increase coincides with the beginning of ripening of sugar accumulation, of fructose and glucose accumulation. Okay? Still with me? Good. Okay, I have 15 minutes still. So here is just a picture of the patterns of malic acid, tartaric acid, glucose and fructose accumulation in the berry. I don't want to go too much into the metabolism of the berry, but just to show how remarkably those metabolic events follow the growth of the cells, follow the change in color, follow the change in other secondary metabolic compound accumulation. And this is what we call developmental regulation. So this is a complex organ that is undergoing a series of events that makes the organ bigger, softer, colorful, and sugar-rich, acid-poor, etc. So you have to think, as a biologist, we say, wow, this is fascinating because you must have a way to control all those processes. And so those processes are controlled temporally, and remarkably, they're also controlled in where they happen in the berry. So, for example, glucose and fructose will accumulate mostly in the pulp. Phenolics, we know, accumulate mostly in the skin. So somehow, those berries can control the metabolism, the synthesis of those compounds, or the accumulation of those compounds, depending on where you are in the berry, if you're in the skin, or if you're in the pulp, or if you're in the seed, for example. And this is also under genetic control. So if you look at two different varieties, we see differences. Here is Cabernet Sauvignon and Sangiovese, just showing the total anthocyanin during ripening, very different patterns, and very different responses in this case to photosynthetic availability. These are when you have 3 leaves or 12 leaves on your vine, how berries respond in terms of phenolic acid accumulation. So you control metabolism in time during development, you control metabolism where it's happening in the berry, and you control metabolism, and I believe the following speaker will focus more on what happens in the berries in terms of tannins, anthocyanin, and flavonol during ripening. But you have also this complexity coming in. We know that flavonols are in the ovary before ripening starts. Then somehow their synthesis is shut down and restart at the very end of ripening. Proanthocyanidins, tannins, tend to accumulate early on during development. And then as a proanthocyanin biosynthesis decline, you kick in with anthocyanin biosynthesis. So at ripening, something must tell the berries to start doing something different. Accumulate sugars, degrade the acids, make anthocyanin instead of other flavonoids. So it's a remarkable developmental control of a series of events. And this is what I want to go to, is how we control those ripening events. The berry must have internal signals that regulate all these events. And this is just a summary of all the hormones that we know accumulate in the berry during development and during ripening. Auxin, this IAA is auxin, declines during development, and we think is a negative regulator of what happens after. Makes sense? We make it before ripening. Now we need to enter ripening. We don't make, or we inactivate auxin, and we can start the ripening process. We know that ABA and ethylene accumulate at the onset of ripening, so we believe that they are actually involved in the activation of the ripening process, and so forth. Brasinosteroids, these are steroids, also accumulate at ripening. So this is just to show that we know something about the signals that may actually regulate all those processes that we care about. And when we think about those processes, you think about berry composition as a winemaker. As a viticulturist, you will think about all the things you can do to those vines in order to improve grape composition. What you are really doing is influences the different composition of those signals that in turn modulate the chemistry that is happening in those berries. And this is, again, a summary slide showing how different hormones may have a positive or negative influence on what is happening inside the berry. So typically auxin, cytokinin are believed to be negative regulators of ripening, whereas ABA, ethylene, and barsinosteroids are believed to be positive regulators that push, accelerate ripening and make, in the case of ABA, for example, make the berry produce more anthocyanin, for example. Okay? So, and this is just to show ABA accumulation, for example, throughout ripening. So this is your bricks in yellow and anthocyanin in black. So both of them accumulate at the onset of ripening. At the same time, ABA spikes. We have an increase of ABA accumulation in those berries. And we believe that this ABA is responsible for some of the processes that result in the ripening, in the beginning of ripening, the sugar accumulation and anthocyanin accumulation. When we treat berries with ABA, and table grape growers know that, we accelerate ripening and we increase color. So if you have color problems, if your color problems because of lack of color accumulation or because of increase of color degradation, actually ABA can compensate. Okay, so we have some ideas about how we control ABA, how we control ripening. And this, just to add complexity and bug you a little bit, sits on top of an even more complicated network of molecules, proteins, that regulate all these processes. So the hormones come in, regulate other proteins, and other proteins will regulate metabolism. And this is just showing the number of these signals, of these regulators, that can be associated with early ripening, with the intermediate, the onset of ripening, the end of development, and with late ripening. So it's very complex. We know something about it. We're still studying a lot of it. And particularly we're interested in how environment affects all these layers of regulation. Again, as viticulturists, we try to fix problems. We try to improve grape composition. What we are doing, we are tapping on these mechanisms of regulations. And that's why we are interested in studying them. And in the lab, we have developed protocols that allow us to link grape metabolism, so organic acid metabolism, sugar accumulation, flavonoids, etc., with all those events upstream that determine those changes in the berry. Okay? And finally, we are trying to link this to flavor as well. And now I'm going to tell you a little bit. I have 5 slides. I know my talk is very long. It's fine. I give you, so I try to explain this complexity of metabolism and developmental regulation. So now let's think about what happens when you have a red blotch in your berries. You know that you reduce the bricks, and you have, sometimes you reduce bricks, you have late maturation of the berries, and loss of color. Loss of color means that you actually are affecting negatively the biosynthesis of anthocyanin. Reduced bricks could be that you have less sugars ending up in the berries. So again, let's move away from what you see in those berries and think about what may happen in those berries because of the presence of the virus. So we conducted a study in the past 2 years that actually tried to understand why berries behave like they do when the virus is in the vine, with the hope that we can find ways, either agronomically or by application of growth regulator, etc., to compensate for those negative effects of the virus on berry metabolism. So we conducted this study actually locally here in Oakville and in Sonoma, and we focused on Zinfandel 3, clone 3 from FPS, and the question we tried to ask was, how does red blotch affect those regulatory events that happen in the berry that in turn result in worsened quality of the berry. And what we did, we collected berries at different ripening stages, pre-veraison, veraison, and post-veraison, from positive and negative vines for red blotch. And we did, I don't want to go too much into the tools we use, but we did all the stuff I mentioned before. All those protocols that allow us to link metabolism with the chemistry of the berry. And so what we observe, actually bricks, we observe a change in bricks in Oakville, but not in Sonoma. This I'm showing here is anthocyanin. So in both sites we had a significant reduction in anthocyanin at harvest. these are the many things, boring for most of you, that we observe, but just to show you, I would just point to a couple of processes. So when we look about berry development and metabolism in the infected berries, we've seen that the virus is causing the expression of those events that are typically associated with the early fruit set, early development of the berry. The virus is inducing them later in ripening. So we've seen Calvin cycle, photosynthesis, all those events associated with green berries happening later in the berry ripening, suggesting that the virus somehow is keeping the berry young. Same thing from the downregulation. So now we are looking at what red blotch actually caused to suppress. We have seen that red blotch suppresses all the good stuff. Suppresses genes involved in the phenylpropanoid pathway, in flavonoid metabolism, in anthocyanin biosynthesis, in tannin biosynthesis, and so forth. And this is just, again, we went into detail. We look at those specific genes that are involved in the pathway and those molecular events that result in a synthesis or not of a certain molecule. And this is the take-home message where we saw a decrease in those metabolic processes that accumulate phenolic acids, flavonols, and anthocyanins. And this is just some examples. Catechin, which typically, as I showed before, proanthocyanin and tannins decline with ripening. There is a significant decline in accumulation pre-veraison of catechin because of the virus. All the anthocyanins we could measure were negatively affected by the virus. So this is not a delay of ripening. This is the virus tapping on we believe the hypothesis is that the virus is tapping on some of those regulatory mechanisms that in turn influence berry composition. So we look at some of those molecules. We look at ABA, we look at gibberellic acid and others, and actually what we found is that at veraison, ABA accumulation is reduced by the virus. Not later on, but at veraison. So somehow the virus, directly or indirectly, is affecting the accumulation of a very important signal for ripening. Okay? And so red blotch reduces abscisic acid at veraison, and we know that it does it by suppressing the key molecule that makes ABA. We don't know how it does it. We don't know if it's a direct effect or indirect effect of the virus. We know that viruses are so-called obligate parasite, so they need to have a young, healthy tissue where to grow. And the ripening is pretty harsh on those organisms because it's a slow death of the berry. You accumulate antioxidant, but you also accumulate oxidation and so forth. So it could be that the virus is pushing against all the processes that actually accelerate ripening and is promoting those mechanisms that slow ripening down. Okay? So the question is here, can we use ABA to attenuate red blotch disease impact on berry composition? So, and this is where we're going now with our research, to try to understand if we can do something in the vineyard to ameliorate, to enhance grape quality before, and save one harvest at least, before having to replant. And that's the final, my final slide. All these data are published and described in a publication from my lab that was released early in the year. It should be freely available to everybody, but if you cannot find it, just email Monica or myself, and I can share it. And so I'm done with this. And so this is. I didn't talk about many of these things, so I don't know. Red blotch work was done with the other faculty in our department, Ibler, and faculty at Penn State and in Spain. And the funding on red blotch came from my startup money, so I don't have to acknowledge anybody there. But yeah, so thank you very much for your attention, and I'll be happy to answer any question you have and doubts and whatever. Yeah, please. Oh.
You showed that ADA was reduced, but gibberellin was reduced as well. Are you guys going to look at gibberellins as well for raising?
So we saw GA reduction in terms of accumulation, but we didn't see other GA-dependent pathways affected by the virus. So that's why we need to focus on something, because we cannot do everything. And ABA makes a lot of sense because ABA is the regulator of the process that we see reduced by the virus. So, yeah.
Thanks.
So in table grapes, ABA is I would say is one of the most recent acquired growth regulator, but it's been applied. It's still not clear yet how to apply it effectively. So in some cases, I believe they soak completely the clusters in a solution. I believe that spraying is also a possibility. So there are some issues about reaching the berries on the other side of the cluster. So if this is a topic application, then you will see color accumulation on one side and not on the other side. That can be an issue. So there is a lot of development, research and development on this, particularly in table grapes. So the slide I showed you, so these, maybe I should not have done this, but bear with me here. This is actually a trial that we were carrying out by applying multiple, was done in Brazil, on table grapes that fail to generate enough color for commercial value. And we were collaborating with this group, applying either one or two applications of ABA to the berries to increase color. So, so this is not, so I don't have any financial interest in pushing ABA applications. And we know there are other hormones involved in color development. So why I wanted to mention this is that we know a lot about berry regulation of these processes. So we may be able to help if we have an open mind. So we could think about brasinosteroids, we could think about etephone treatment. Etephone is a molecule that releases ethylene as it is applied to a plant, and it is used in horticultural crops very often, is commercially available. And we know that ethylene, if applied at the close to ripening, actually pushes ripening. Is not directly linked to color, but accelerate ripening. So we know something that we could apply to the berries to improve their composition. And yeah, so yes.
I think some of the early work on red blotch suggested that the leaves were also reduced in photosynthesis, and maybe that was partially responsible for decreased Brix accumulation.
Yes. Yeah.
Are there going to be other pinch points in being able to get increased Brix?
So sugar is very interesting because sugar is what you value as Brix and for winemaking, but it's also a very subtle signaling molecule. So it could be that by affecting sugar delivery into the berry, you're also in turn compromising some of these pathways. Yeah, we know that ABA and glucose in particular go together. ABA induces some of the proteins that are related to glucose and with feedback regulation. So there is a lot of overlap. I don't touch leaves, so maybe Dr. Cortural will have something about photosynthesis, but clearly it's all connected, particularly for sugars, because they're not made in the berries. So the source must be affected by the virus. And anthocyanin, tannins, they're all made in the berry. So that's why allow us to look at their biosynthesis directly in the berry. But the caveat of that is that the starting point of anthocyanin biosynthesis, everything is glucose as well. So which could have again effects on downstream, which we are overlooking here. Yes.
So again, in table grapes, they use potassium metasolate to push sugar into the berry, but again, that was related to ABA biosynthesis as well.
Yeah, it's all connected. The beauty and the complexity is all connected. So yeah.
You had one slide, a couple slides back, that showed a berry that was half purple and half.
Yeah.
And I'm just wondering if you could comment on why there's a question mark next to it. Is it steroids? Because there's a lot of seaweed-based sprays and products out there now that are claiming.
So the question mark is actually the link between auxin and bacillinosteroid. This is not a question mark. We know bacillinosteroids are positive regulator of ripening. So they are effective. We know that they accumulate in the berry normally, and we know if we apply them to the berry, we induce ripening. So this is established. The question mark is the link between auxin and bacillinosteroids. Whether auxin is a negative regulator or also bacillinosteroids. Okay? So yeah, I should have removed that because we don't know anything about it, so we should not talk about it. But I got this from another group, and yeah, and this is not a real berry, okay? It's a collage, yeah. Question?
Is there a point at which you could apply cytokinin inflorescence formation versus vegetative growth in advance?
That's a very good question. And I don't know, I know experimentally it's been done and was successful, particularly because tendrils can be converted into cluster even later during development. But I don't know from a commercial point of view. And I don't know, when you apply these molecules, it's always, you need to think where those molecules end up. As I mentioned before, your GA treatment, if it ends up on the buds when they are undergoing differentiation, then you may actually reduce differentiation, while you are positively inducing growth, but you're still compromising year 2. So when you think about hormones, there are not so many in plants, and they do many things with even opposite effects. So where you apply them, when you apply them, how much, and is very critical. So yeah.
Was the research done on the cytokine application in Davis, or where,
Ah, wow, I know papers from the 70s, so before I was born, and I don't know what. I think some of them are, I believe, are from Cleaver. Cleaver, yes, from Davis, some of them, yes. Yeah, yeah. Yes?
What about the impacts of cultural stuff, hedging, leafing, lateral removal on the hormones and the time of it? Besides getting light in there, looking at hedge versus untrimmed, or hedge versus unhedged lines. Is there an impact on that as far as ripening goes?
I would say, yeah. Mostly from what I showed today, mostly on light and exposure of the berries to light, and then obviously when you are affecting the source of sugars, it will affect all the downstream events that happen in those berries. But from a standpoint of secondary metabolism, we think that light is a major factor, and I believe Dr. Cultura will talk about shading treatments today.
Stay around it.
Yeah.
I was worried about hormone.
Yeah, but it's all linked again. So we know that light and ABA are connected, light and ethylene are connected. So yeah, so when you think about hormones in plants, hormones and other signaling molecules are the interface between the environment, between viticulture and the vine. So whatever happens in the vine is because of changes in their signals. So this does not mean that the application you make exogenously reproduce what happens inside the vine. So you may actually, when you put a lot of ABA, you may trigger pathways that would not be active otherwise. So it's, we apply parts per million in plants. These are active at picomolar, nanomolar concentration. So, but I like to think that this molecular viticulture is actually what explains all the things we do in the vineyard in terms of the changes we see in the berries. Is that interface between viticulture and composition. But, yeah. So again, it's all connected. So we know something, we don't know everything, but I believe that it's all connected. Yeah. Good. Okay, thank you for the attention.